these ways of existing. If it is integral, like, have you ever heard the saying, they were an integral part of the committee, for example, okay? An integral part of the team, okay? Important, embedded, inside, okay? So, an integral protein, they're very deep in the membrane. There's only a tiny portion that might actually show on that outside of the plasma membrane and the inside. There might not be a whole lot that's outside of the plasma membrane on either side. So they are embedded, like almost to the same height as a phospholipid bilayer. One of the jobs that this type of protein can do is they can be a channel. Now, let's go back a picture. Some of these proteins can be a channel. Okay? An extrinsic or peripheral. We can see that there could be something that they're connected to, either inside that plasma membrane or outside. Now, their function. What did we say about proteins? Huh? What? Y'all gotta speak up. Say it again. They do the work. And structure determines function. Okay? So no matter what protein we talk about, structure always determines function. So their function is going to depend on their shape. Any other type of characteristics, okay, because proteins are coded for by my DNA, a gene on my DNA, and they can function to be a marker. So we could see that protein, and it could have something on the outside of that phospholipid bilayer, and it's a marker. It's going, woohoo, kidney cell, woohoo, liver cell, woohoo, intestinal cell. Okay? It could be a marker. It could be a point of attachment. Receptor or another cell, attach. Come here, we need to hook together. Okay? They could be a channel. They could allow something to move. The receptors, they could actually be an enzyme. It could be there in between on the outside, where on the extracellular fluid, there might be it could be a molecule that if it's out here in the extracellular fluid, it can move into, and that protein will act as an enzyme to break it down so those materials can move into the cell. They can be carriers. They can allow something to attach and let it move into the cell. So we can have materials out here, but the only way it's going to get into the cell is if there's a protein that lets it. Not every particle in the extracellular fluid, out here, in this extracellular fluid, we could have amino acids, we could have sugars, we could have oxygen, we can have hormones. Do you see where I'm going with this? Everything the cell might need, hopefully, is in that extracellular fluid. It's up to the cell itself to get that material. Some of the material will and can just simply glide through the plasma membrane. But that's not a lot, okay? A lot of times, the majority of the materials that a cell would use, we need a protein. So, let me think here. Channel proteins, marker proteins, receptor proteins, enzymes, what is that telling us about the number of proteins that can be present in that plasma membrane? We're going to find that we have them in different categories and more than one exists. Does that make sense? Okay, so just right now, at this point in time, what are you learning about the importance of the plasma membrane? We're definitely getting a lot of control. Definitely. Anybody else?
Its function and why it's important.
I think it's, see, this is why when I hear people talk about the nucleus being the most important part of the cell, I argue with them. I'm like, whoa, hold up. Let's talk about the plasma membrane for a second.
It's good. Like, it's when you like taking bio, they always like they like emphasize it. They don't really never talk about the plasma. So everybody's always like nucleus is. That's it.
And that's because usually in the general bio class, the goal is really, well, let's see about the parts of the cell, and then they tend to emphasize the DNA. And of course, that's the part that's in the nucleus. Once again, don't get me wrong, that's important because, hold up, let me think about this for a second. I now see that I got a ton of these proteins in this plasma membrane. What is it that codes for these proteins?
Yeah.
The genes on my DNA, which are found in the nucleus. So it's still important, but that DNA wouldn't get to function if the plasma membrane wasn't doing its job. So just something about it. So these proteins, we now see that they can be like almost the same height as the phospholipid bilayer itself, or they can be extending inside and outside and doing all these other jobs that need to be done. So, if a protein is acting as these glycoproteins, meaning they have the glucose chain attached to it. One of the jobs that they will do is they can act like a marker. Meaning, let this cell be identified. So anything that might be out there in that extracellular fluid, that glucose chain that's existing is saying, this is me, this is my identification. So it's important in immunity. I don't want my own cells attacking my cells. Ever heard of an autoimmune disorder? Okay, this is where things could go wrong. I need recognition. The cells need to be able to recognize each other. A good example, egg and sperm. Okay, the sperm needs to know or be able to identify which cell to try to enter. So we could have, they need to be able to recognize each other. My cells need to communicate. For example, if we get exposed to a virus and that virus invades one of my cells, it works really hard to let the neighboring cells know it's been invaded. And then, hopefully, immune system kicks in and helps with protection. But in the case of like a virus, for example, the cells want to communicate. The one that got invaded wants to say, watch out, there's a foreign object in the extracellular fluid, and it's going to try to affect you. So we can have them in acting as a marker, the identification that needs to take place. Proteins that can be extending, like outside that plasma membrane, have a little bit of an extension off of itself. They can be points of attachment. It's important to note that as animals, as humans, we are very well organized. Our cells organize into the tissues that make up the organs of the organ systems of this organism. We are very organized. So they can act as points of attachment, allowing cells that need to be together to attach together. We can see that if they are very well embedded within that plasma membrane, they can act as a way of communication, but this time inside the cell. Not every substance that makes it to a protein will actually enter the cell. It may just simply come to a protein all right? It may just be a substance out here, and it just simply attaches to that protein, but because this protein has another portion